Welcome back to The Explainer. Today we're tackling what is honestly the ultimate organizational challenge in the history of the world, classifying life itself. I mean, just think about the sheer chaos out there in nature. You've got billions of organisms, ranging from literally invisible microbes to massive blue whales. How do we even begin to make sense of all that? Well, biologists had to build the ultimate filing system. And the story of how that system evolved, it's absolutely fascinating.
Okay, let's dive into this. Our roadmap for navigating this biological filing system hits the quest to classify, Whitaker's 5 kingdoms, the bacterial world of Monera, the microscopic misfits of Protista, and finally, the great decomposers, the fungi.
All right, starting off with Section 1, the quest classify. Let's look at those early systems.
Now, since the dawn of civilization, humans have basically instinctively categorized plants and animals. I mean, we had to, just to survive, knowing what we could eat, use for shelter, or wear. But applying strict scientific rules to this whole process, that took a bit longer. In ancient times, Aristotle tried sorting things using super simple physical traits. Plants were trees, shrubs, or herbs, and animals, well, they either had red blood or they didn't. Fast forward to the 18th century, and Linnaeus gave us the two-kingdom system, Plantae and Animalia. It was easy to understand, for sure, but it was highly inadequate. It lumped together vastly different organisms, completely failing to distinguish between single-celled and multi-celled life, or even between things that make their own food and those that don't. We desperately needed a better system, which brings us to 1969,
which lands us in Section 2, Whitaker's 5 kingdoms, the modern system. So R.H. Whitaker stepped up and completely revolutionized how we file away life forms, solving all those major headaches caused by the old two-kingdom setup. Before we even talk about plants or animals, Whitaker realized we had to look closer at cell structure. Prokaryotic cells are the simplest, they don't have a nuclear membrane. Eukaryotic cells, on the other hand, possess a well-defined nucleus and these complex membrane-bound organelles. And honestly, this single distinction right here, that is the absolute bedrock of modern biological classification. By mapping out criteria like cell type, cell wall composition, and how these things get their nutrition, Whitaker created 5 distinct biological folders: Monera, Protista, fungi, Plantae, and Animalia. And it brilliantly illustrates how this solves the old problems. Instead of forcing literally everything into a plant or animal box, we now have specialized categories that reflect true evolutionary and structural relationships. It makes so much more sense.
To really understand why this overhaul was so necessary, just think about a mushroom. Under the old system, fungi were tossed in with plants just because they both had cell walls and, well, didn't move around much. But look at the structural difference. Plant cell walls are made of cellulose, while fungi have cell walls composed of a super tough substance called chitin. Plus, fungi don't even make their own food. This stark contrast shows exactly why Whitaker had to carve out a brand new kingdom just for fungi, and this matters immensely in the real world. That fundamental difference in cell wall structure, that's exactly how certain antibiotics target and destroy bacterial cell walls without harming our own human cells, or affecting fungi for that matter.
Moving on to Section 3, Monera, the bacterial world.
Let's open up the very first of Whitaker's filing cabinets. Welcome to the invisible, extreme, and incredibly abundant realm of Monera. Bacteria are grouped by their distinct geometries. You've got the spherical cocci, rod-like bacilli, spiral spirilla, and the comma-shaped vibrio. Now, they might look remarkably simple, but do not let that basic appearance fool you. While their physical structure is straightforward, as a group, bacteria boast the most extensive metabolic diversity of any living things on Earth. They are the ultimate survivors. The staggering variety within Monera is literally mind-blowing. First, you have archaeobacteria, the total extremists, living in ultra-salty waters, boiling hot springs, or even acting as methanogens in the guts of cows, naturally producing biogas. Then there are the eubacteria, the true bacteria. This group includes cyanobacteria, which can form massive blooms in polluted waters, and those incredibly vital heterotrophic bacteria that decompose dead matter, or, cause devastating diseases like cholera. And we absolutely cannot forget mycoplasma. These are the smallest living cells known, and shockingly, they survive without any cell wall at all. Just wild.
So, how do these abundant organisms sustain such massive populations? Well, the crucial point is that they primarily reproduce via fission. It's a simple, rapid splitting of the cell that lets them multiply at absolutely astonishing rates. And when conditions get incredibly tough, they can just produce these hardy spores to survive until the environment finally gets better.
All right, let's turn to Section 4, Protista, the microscopic misfits.
Moving up the ladder of complexity, if Monera is for the prokaryotes, kingdom Protista is essentially the designated filing cabinet for all single-celled eukaryotes. Honestly, it's a bit of a biological junk drawer. I say junk drawer because the boundaries of Protista are notoriously blurry. What's a photosynthetic protistan to one biologist might literally be a plant to another. Because of this, it forms a fascinating, somewhat messy link with plants, animals, and fungi, placing some incredibly strange bedfellows right next to each other.
Just consider the staggering contrast within this single kingdom. On one hand, we have dinoflagellates, heavily armored with stiff cellulose plates. These are the organisms that multiply so rapidly they cause those toxic red tides in the ocean. But then, on the other hand, we have the euglena. It lacks a cell wall entirely, possessing a flexible protein layer instead. It's photosynthetic in the sun, but if you put it in the dark, it suddenly turns into a predator, hunting smaller organisms. Same kingdom, completely different lifestyles.
And the wild extremes of Protista just keep going. You have crucifers like diatoms, whose indestructible silica cell walls pile up over billions of years to create gritty, diatomaceous earth. We actually use that in real life for polishing and filtration. Then there are slime molds, which literally creep along decaying twigs and merge into these giant aggregations called plasmodium. And finally, the protozoans. These are animal-like predators and parasites. This group includes the amoeba and, infamously, Plasmodium, the parasite responsible for causing malaria.
That brings us to section 5, fungi, the great decomposers. This kingdom is a cosmopolitan, chitin-walled world that touches our daily lives in way more ways than you might realize. Everything from the annoying mold on a piece of old bread to the life-saving antibiotic, penicillin. Fungi are unique heterotrophs, and their lifestyles generally split into two main camps. Saprophytic fungi are the ultimate recyclers. They absorb soluble organic matter from dead substrates, basically keeping our world clean by decomposing forest debris. Parasitic fungi, however, depend entirely on living hosts, and they can be devastating, causing severe diseases in plants like wheat rust or serious infections in animals.
Now, when it comes to reproducing sexually, fungi follow a very specific three-step dance. First is plasmogamy, where the protoplasms of two gametes fuse together. Second is karyogamy, where the actual nuclei fuse. And third, meiosis, which results in new haploid spores. But here's where it gets incredibly weird. Some fungi experience a delay between steps 1 and 2, resulting in a dikaryotic phase. That means for a while, a single cell is just walking around carrying two distinct nuclei. How crazy is that?
Scientists sort these fungi into 4 main groups based on their structure and how they produce spores. We have the phycomycetes, like your classic bread mold, the ascomycetes, or sac fungi, which includes baking yeast, the basidiomycetes, which are your classic mushrooms, bracket fungi, and puffballs, and then there are the deuteromycetes. These are often called imperfect fungi because they act as a temporary holding pen. Scientists put fungi here when they've only observed their asexual reproduction. Once a sexual stage is finally discovered, boom, they get formally moved to one of the other classes. Let's zoom out for a second and see how all this builds into something totally recognizable. All those complex Latin names and microscopic spores eventually give us something like agaricus. That's the common, edible mushroom you might find in your grocery store or growing out in a damp forest. It belongs right there in the Basidiomycetes class, acting as the fruiting body that spreads spores out into the wind.
So, what does this all mean? If bacteria are the extreme survivalists, protists are the microscopic misfits, and fungi are the world's great decomposers with their own bizarre sexual cycles, where exactly do we fit? Where does a massive redwood tree or a human being slot into Whitaker's ultimate biological filing system? Well, that massive complexity is reserved for kingdoms Plantae and Animalia, and those are incredibly vast topics we're going to have to explore next time. Thanks for joining me for this explainer, and keep questioning the incredible structure of the world around you.